All right, I'm going to move us right along to our second to last presenter, Will Miranda, who was mentored by Priya Desarma. If you'd like to introduce Will.
Absolutely. So Will is a phenomenally talented bioartist. I don't know if I've heard that term before, but certainly it applies to Will, with whom I've had the joy to work with this summer. So we work on astrobiology, which is a little bit different from most of the projects we've heard from so far. And when we started, Will and I set goals that Will wanted to achieve, including identifying resources, learning how to read and interpret literature. And Will did a great job in not only taking their own artistic talent, but also this amazing body of information and visualizing very abstract concepts, including life in extreme environments, including archaea, which Daniel might be interested in, as well as their organelles, which prokaryotes were never believed to have had until they were discovered in these organisms. And so Will had to cover a range of topics, including genetics, proteomics, a lot of jargon, biology, and chemistry, all sorts of things combined, and took all of this body of knowledge on these novel nanoparticles, which have astrobiological significance, which you'll hear about in just a moment, and interpret it in such a way that it becomes more outward facing, public facing, and interpretable. So I will not say any more. I'll let Will share more about their work now. Thank you.
Thank you. So as Priya mentioned, I was working on astrobiology. Specifically, I was working with haloarchaea, and more specifically, I was working on this type of organelle that they produce called gas vesicle nanoparticles. So why is this not going? Okay. So haloarchaea are prokaryotes. They're extremophiles, meaning they live in extreme climates. And haloarchaea specifically live in hypersaline, or very concentrated with salt water, but they can also resist many other extreme conditions. Their adaptations would allow them to survive on many other planets that we found, so they could potentially exist, or similar things could exist on other worlds. Gas vesicle nanoparticles are prokaryotic organelles. You find them in bacteria and archaea. We're focusing on haloarchaea. They have an incredibly stable lipid-free protein membrane, which is unique in nature, and that membrane is not liquid soluble, so it doesn't allow the liquid of the cell inside, but it is gas soluble and filled with gas that basically matches atmospheric composition. When they're purified, which is just when they are removed from the organisms they grow in, they can be used for a variety of medical applications. These applications all pretty much hinge on being able to attach fusion proteins to the outside of GVMPs. I'll cover what that is later. Because haloarchaea can survive in so many adverse conditions, it would be possible to take some into space and grow the gas vesicles there, making them useful for in-space medicine. So over here we have one of my drawings of a gas vesicle nanoparticle, and you'll see in pink with the white spiral ribbing, that's GVPA. That's the main structural protein of EVMPs. And then in spirals attached to it, you have GVPC, which is sort of the second most important structural protein. There are many proteins, I think 13, yeah, 13, involved in GVMP synthesis. And so these attach to GVPA along the spiral ribbing. So my project was a bit different from most other people's. I was creating educational materials for a website called HaloEd. It's a great resource on haloarchaea mostly, but also biology in general, a great educational resource. And I created text and images about GVMPs to add to the website. I can't fit all the material I covered into this slideshow because there's a time limit, but this is an example of something that I had to cut for time. This is a representation of the two operons in a specific strain of haloarchaea, NRC 100. Well, no, the NRC 1, and then the plasmid is at PNRC 100. The leftward operon is blue, the rightward operon is purple, and I'm going into more detail than it's fine. Whatever. So I had two main objectives in creating my materials. First of all, I wanted them to be comprehensive, covering everything important to basically understanding what GVMPs are and what their utility is. But I also wanted them to be comprehensible and easily understandable, even for people who don't necessarily have the strongest background in biology, which I believe I accomplished both. So there were two main phases of my project. The first was research. I read through basically every publicly available article on GVMPs in the National Library of Medicine, including an encyclopedia article. And then I wrote out and drew the educational materials to combine all that information into an easily digestible format. I did the original drafts in my sketchbook so I could have my text right next to the images that accompanied them. And then I finalized the materials and threw them into a Google Doc just to have them online. I did touch-ups to the images in Krita and Photopea, and one of my final drawings was done entirely in Krita. That's on this slide. And so I don't have traditional results, but I have product that I created, and it's a new section for the HaloEd website. As of now, the materials are finished. They have not gone up on the website yet. They will soon. So I'm just going to do a quick overview of some of the medical applications of GVMPs. So what we have here is a drawing of GVMP with GVPC on the outside. And then what happens in this drawing is that more GVPC is added to the outside. And this can happen because wild type or just naturally occurring unmutated GVMPs are not fully saturated with GVPC. Adding GVPC on is one of the ways that we can attach fusion proteins to GVMPs. What are fusion proteins? Fusion proteins are basically engineered proteins where you take a segment of DNA that codes for one gene and a segment of DNA that codes for another gene, and you chop them up and you splice them together, and so they code for a fusion protein. And ideally, this fusion protein is going to successfully have characteristics of both proteins. So on the screen, I've got one example of how you can use fusion proteins for GVMPs. So this is to attach antigens, which can be used for therapeutic purposes if your body's not producing the antigens you need to fight off disease, for instance, for immunocompromised people. And so we have the wild type GVMP, and then in this case, the fusion proteins were replicated in E. coli, and you have the fusion proteins which take part of a GVPC protein, which allows it to bind to the outside, and part of an immunoglobulin binding protein, which is what basically every antibody we use for these things attaches to. I don't know about exceptions, but those are fused together, and then they're attached to the outside, and then you can attach antibodies to this, and you can see, as illustrated here, you can attach multiple different types of antibodies, potentially multiple different types of antibodies on one GVMP. And there are a lot of other things you can do. For instance, you can just attach antigens, which are the things that antibodies target and recognize. So you could use them for vaccines, and you could use them for so many other types of proteins. For instance, there's this one protein that protects against septic shock, which is a type of endotoxic shock where basically your immune system goes all haywire and it starts causing inflammation everywhere, and it's very deadly. Even with being hospitalized, the mortality rate is between 28 and 40%, and it can be treated with a certain protein, but the problem with that is it doesn't have a very long shelf life, and it needs to be stored in the cold, which if you attach it to a GVMP, it's very stable, it doesn't expire as quickly, and it doesn't necessarily need to be stored in cold. And there are probably so many other applications you could use them for, so they could make a really big impact on medical technology. The significance of my project was that almost all previously available information on GVMPs was found in scientific papers, which are great, but they're very hard for most people to understand. This project has taken information from over a dozen such papers and compiled it into one place in a way that I believe even a layperson could understand. So here at the end, I have basically a gallery of some of the images that I drew, which did not make it into previous parts of the slideshow. So I'm just going to show them here. This is one of my favorites. It's also the first one I did. This is another of my favorites. This was not for my work with Halorkia and stuff. This was for the Swift satellite team's 20th anniversary of their launch. They wanted some drawings. This is the first one I've done. There are going to be 2 more. This is not the final version.
Yes, I reached out to Will because I knew she was such a great artist. Because Swift really wanted somebody to be able to make the satellites look like they were friendly and they had friends and they were all working together to study something. So this is what she came up with, and I just think it's so cute.
I use they them pronouns, but thank you so much.
Oh, sorry. Sorry
Yeah,
for
no,
that.
no problem. And these are my references, my sources. So I would really like to thank the N3 program so much for this opportunity, and I'd especially like to thank Priya, Disarma, and Disarma Laboratories for all the help and the guidance and just making this an amazing experience and guiding me through, but also letting me learn on my own and just being available whenever I needed clarification or help understanding something. And it's been great, and I'm so grateful. And thank all of you for watching.
Thanks, Will. Amazing artwork. Getting a lot of praise in the chat about that.
I
And-
can see a future in art there, bio art. We need people who can actually translate, you know, those computer generated images, as much as that you guys like it all, but to see it in such a beautiful fashion, so clear. I think Will has a career ahead of themselves.
Yes, and also, you know, I mean, if you don't think it's a career, there are lots of people that work for NASA that do this kind of artwork. Not necessarily towards biological things, but sometimes, you know, sometimes spacey things, which is why I'm glad that Will got the chance to draw some satellites as well. And as you know, here at Edion, we have a full-time artist that has been working in my group for 25 years, drawing all sorts of spacey stuff. So yeah, it's actually a real career that you can have. So, and I think Will is well on their way to do that. So just congratulations, just wonderful job.
Amazing. You can take one question for Will. We still have one presenter left. If there are any. So you can always follow up with them in the chat as well. Okay.
Will, did you want to mention how you can transport these microbes into space inside of what?
Oh, in salt crystals. So a lot of the times they live in these tiny water pockets in salt crystals. They're, again, very, very high concentration salt. If you put them in, say, the ocean, they die so quickly because it's not concentrated enough salt. It's very nearly saturated salt. So you put them in salt crystals and they can live in there for decades and stuff. And you could take those into space and grow them from scratch.
Thanks for sharing.